Good to go. Okay, awesome. Well, this is so exciting, everyone. This morning is not quite our official kickoff. Tomorrow morning, we have some welcomes from our sponsors through NIFA and FR. But this morning, we get started. Today is our training day, and one of our three objectives for the International Weed Genomics Consortium is to have training and networking, and that's really what we're achieving here at the conference today. So welcome to Kansas City. For everyone who's here and to everyone virtually, welcome and thank you for joining us. It's a great pleasure for me to introduce our training speaker for the morning. We're welcoming Koen Nijbroek. He is with KeyGene in the Netherlands, and KeyGene has developed an online database and platform for genomics called CropPedia, but in our case, we'll be calling it WeedPedia, of course, Weedomics. And this morning, Koen will be taking us through the functions of CropPedia, so you can understand how to search it, how to work with the different features for the genomes that will be uploaded over the course of now and the next couple of years. One thing I'd like to make sure everyone knows is that you can type in questions for Koen on Chime. So when you're in Chime, you'll see a function called Q&A, and that's in the lower left-hand corner. So please, at any time when you have a question, type that in, and then when we have the breaks, that's when we'll read those questions and get answers for you. So please don't wait until the end. You can type in questions during that Q&A time as well, of course, but there'll be just a little bit of a delay. So I encourage you to type in your questions as you think of them. And then for everyone in the room, we'll have the microphone for you so you can ask the questions as well for our speaker. So with that, again, it's a real pleasure to have you here. So excited to see everyone. And we will turn it over to Koen for our KeyGene training session. Thank you.
Good morning, Kansas. Nice to be present here. My name is Koen Nijbroek, born and raised in the Netherlands. Currently working for KeyGene, a company based in Wageningen. I will guide you through CropPedia, WeedPedia by presentation / workshop. So the way that this morning will look like is I will start with giving a general introduction about the core functionalities of CropPedia, then switch over to the pedia that you are probably most interested in, the WeedPedia. And since the WeedPedia is fairly new and still fairly empty with data, we will give a live demonstration by a rice use case. And then later on, I will also in detail explain about all the different types of data that you can put into CropPedia and how you can visualize that. So there will be breaks and Q&As in between, so don't hesitate to ask any questions. First about KeyGene. KeyGene was founded in 1989 by 4 Dutch seed companies. And even though they are all competitors to each other, they decided to combine their forces and create KeyGene to do a lot of science there. And we define ourselves as the go-to ag biotech company for higher crop yield and quality. We specialize in crop improvement by molecular breeding. And one of the tools that we made is CropPedia. And unfortunately, I only am present virtually. I was in Kansas City 2 years ago, but due to COVID, I was not able to attend physically. Nevertheless, this should be more than fine. So, since a lot of data has been generated proprietary as well as is present publicly, there is a need to combine all that data together and to interfere all the data for your genomic and genetic research. and that's where KeyGene comes in. And KeyGene is a single system that can combine pretty much every single type of genetic and genomic data. And then you can think of gene annotations, variomic data, RNA-seq data, proteomics, metabolomics, and it even includes PubMed literature as well. And this is how it looks like from the front page, and we will go into the Pedia later on, but I think it makes more sense to explain the core functionalities first. It's a, again, single system where all the data is stored into, and you can interfere with the data by 6 main functionalities. And we will go through each and every single functionality in more detail later. But the 6 functionalities are Genome Browser, Genetic Map Viewer, Variomics Viewer, a Data Browser, a Local BLAST tool, which has the same capabilities as the NCBI BLAST, but then the advantage is that you don't have to do it online. And it includes functionalities for omics analysis. So the home screen will basically guide you through each and every single one of these functionalities. On the right pane, on every single page, you can find an extensive menu where you can also browse through these functionalities, but where you can also find extensive help sections. So if anything is unclear at any given moment, you can just have a look at the help. There's an extensive search interface. So the more data you put into KeyGene, the more results you will get. And if you are working with a specific crop or a specific reference genome of a crop, you might want to enable or disable certain data sets. So with advanced searching, you can basically decide what you want to retrieve from the database. And as you see here, there's an example of a Solanum database. There are a couple of varieties in there, like Lycopersicum, Habochitis, Pinelli, Arcanum. If you're only working on Solanum Lycopersicum and you're only working on the latest, well, it's actually not the latest genome, but version 3.0, then you can just enable 3.0 and disable all the other databases. Once you hit a search term in KeyGene, you will basically see a window that will come up with all the results that have been found of all the data that is in the system. And that can be a lot. So if you type in a generic search term as flowering or yield, every single data component in the system that contains that exact word will be shown back. And we will go into the details of this search later. But on the left top pane, you will see gene passports. And basically, that's the same as a passport that each and every one of us has for traveling. It contains all the essential information about a specific gene. We also have a pane for genomic features, overviews, genetic features. You will see a pane where on a genome all your results are located. So if you are doing some specific research, you might end up that all your search terms are roughly located at the same genomic locus. There is a link with PubMed where you can browse for literature. And at the bottom pane, we also have pathway information whenever relevant. One thing in KeyGene that you can do is make a digital notebook or lab journal. So every single data entry that you find in KeyGene and that you might find interesting for your scientific research, you can save it in a workspace. And basically, you can give it the layout and structure that you desire. You can even share it with other colleagues or users or generate extracts out of that for other purposes. So the main core functionality of KeyPedia is the Genome Browser. And I hope that most of you are familiar with the concept genome browsers. It's widely used in the public domain as well. And basically what it does, a genome browser is a representation, a linear representation of a genome. And you can visualize all kinds of properties of that genome. A genome browser is limited to only be able to view one genome at once. So if you are interested in multiple genomes, as we see on the screen here, you have to open multiple genome browsers. So this is what it looks like. This is obviously the genome browser tailored to KeyPedia, but it resembles quite well with common genome browsers in the public domain. So on the top pane, you will find navigation features that will allow you to go to certain positions or regions in your genome, to zoom in or zoom out the region that you are looking at. And on the left side pane, you can enable what we call tracks. So every data file that goes into KeyPedia is called a track. You can enable or disable that track to see the data that is in there. If we zoom in a little bit and only look at a single gene, for example, you can see the whole genomic region. So you see that it obviously starts with HEG. You see the different reading frames that are possible. You see the protein sequences that are possible. And you can then look into details at features. So if you would select this particular gene, which was done on the right bottom, then you can see that you have multiple actions that you can perform on this specific data feature. You can save it to the digital notebook that I just mentioned. You can download sequence. You can search for it in KeyPedia to find other relevant hits. You can pull up that passport, which contains all the essential information about the gene. And you can download also kinds of other data types. So going into the gene passports, as mentioned, it contains all the essential information that a gene might contain. And this is not data that can be easily obtained from the public domain. So data that you download from the public domain is usually limited to a certain aspect, whereas gene passports try to combine all different kinds of aspects. So you need to either run a sophisticated gene annotation pipeline or try to combine all kinds of data together. But if you have that data, what you can do is look at accession information, look at predicted localization, interpro results, BLAST annotations, GO accessions, transcription factor information, phylogenetic trees, protein clustering. But we will go into this in detail more later. So this is the search bar again, and also from there you can browse to the gene passports. Also from the Genome Browser, you can browse to the gene passports. So all the data in KeyPedia and all different ways of approaching the data is also linked to each other, so you can easily navigate to specific features. In KeyPedia, you can compare genetic maps. So if there are genetic maps available of the crop that you're interested in or the genome that you're interested in, usually you will see that genetic maps from the public domain are relatively limited in the number of markers that are present. But if there are a variety of different genetic maps, you can easily compare them and see whether markers are in the same orientation or have the same physical position. So that's what you see here, where we enabled a genetic map and linked it to another genetic map, and then linked it to another genetic map, and so on. And you can see here all the connections where similarity between the 3 genetic maps were observed. Here's a closer look into that. So you see, for example, here that there's one match between these 2 genetic maps, but then none after that, whereas another genetic map, which is not observed here, contains far more, many more matches. KeyPedia also has a BLAST functionality. So it's KeyPedia is mainly intended for visualization and data interrogation, not necessarily data creation. So running very computationally intensive BLAST jobs on KeyPedia is not recommended. For that, you probably would ask a bioinformatician to do a BLAST for you and then visualize the result of that. But nevertheless, you may have some proprietary sequence information that you do not want to put into the NCBI database, or maybe it's not the policy to allow for that. So for simplistic BLAST jobs, you can do it in KeyPedia locally, so your proprietary information is therefore protected. It contains all the same functionalities as what you would do with the NCBI BLAST. So you can choose for the different types of algorithms. You can go into the advanced settings and change values whenever applicable. So the results of that will be only visible for you once you are logged in. You can again save it to your digital notebook, display it in a Genome Browser, and from there you can also again share with other colleagues. And here you see an example BLAST job, and hopefully you're all familiar with the output of BLAST. If not, we will go into detail in the last section of this demonstration. But here you can see that we found a match on the Solanum lycopersicum genome chromosome 7. Another functionality in KeyPedia is the Variomics viewer. So if you have a lot of resequenced lines or any other type of resequencing data that's often stored in a variant calling format, VCF, also that we will discuss in detail later. But there is a specific functionality for that that can load VCFs and display them. And from there you can very easily search for patterns or functional SNPs. You can also display it in the Genome Browser, but the Variomics view is a bit more detailed and extended for displaying variant information. So this is how it looks like. On top, it's the same linear representation as a Genome Browser. And then on the vertical axis, you see the different accessions that are in there. And the more intense the color of a specific block is, the more variation has been observed. And you can already, this is just a view of all the variation of all 12 chromosomes of, in this case, lycopersicum. You can already see that there are a couple of accessions that have the same SNP patterns, whereas there are other accessions that do not have this specific pattern. So if you're interested in, for example, a group of lines that are resistant to something or show a specific phenotype versus a group of lines that do not, there you can easily make these visualizations and search for patterns or SNP annotations. So there is a extensive way of narrowing down that whole view of the genome and to search for variation of interest. You can make contrasts. You can apply filters to how variation may behave over a certain amount of lines. There is, if available, SNP annotation, and then you can filter on the impact of variation. You can also filter on the quality and the depth for retrieving variants that are more reliable, for example. So that's the core functionalities of WeedPedia. So now we will go to the actual WeedPedia, have a look and feel of how WeedPedia works and operates, and see what is already available for you. So this is the WeedPedia website, weedpedia.weedgenomics.org. And I will for now use a KeyGene login. But if you have a generic account, then you are able to log in and view all the data that is available for that generic account or for you. So there is a lot of proprietary information in this WeedPedia as well, which is specific for certain individuals. And you will not be able to view or extract any of that data. You can only see the data which has been assigned to you. And as a general user, you will be able to find two different types of data sets in the WeedPedia. Passia scoparia, which is, of course, a type of weed. And the other one, and I think the common name is horseweed. There are two genomes available in the WeedPedia for all. And there is, at least for one of the genomes, already some gene annotation available. But we will see that later. So now this should look familiar. We have these 5 core functionalities: Genome Browser, the map viewer, the Variomics viewer, BLAST, as well as omics analysis. You can make workspaces, and we will do that later with the RISE demonstration. And on the top hamburger menu, you have different options. We have these 5 core functionalities again, but you can also opt for the help. And if you click the help, you will find an extensive section that will tell you everything about the most important window of WeedPedia, the homepage. And it will explain everything in detail much more than I will do now. So if we have a look at the weed data that is already available, if we have a look at the first genome, then you will see 2 different tracks available in the WeedPedia. So the first is the reference sequence, and the other one is a genomic feature, which contains gene annotation. So on the top pane, you can browse through the genome of this weed and have a look at the regions or genes that you are interested in. You can either browse or navigate to areas, but you can also type it in. And if you have very specific search terms or names of genes, then you can also just enter it in the search and then go to it directly. You can zoom out and zoom in, obviously. However, the more you zoom out, the more data will be visualized. And you can already see here that the max height has been reached for this specific track. So there's a lot of data available still here, but you're not able to see it because you're now looking at 1 MB of sequencing content. So that's a lot. There are multiple ways of tailoring your visualization. So every single track has this small button next to it, and then you can edit the configuration or the way of displaying. So if I would opt to collapse this track, then everything will be condensed in one single entry. But then you also lose a lot of data that you would otherwise see on the screen. So it depends on how you want to visualize your data and what you want to see. But in a normal situation, looking at a very large area, it's simply too much to display. So there is a way, of course, to zoom in, either via the top panel or by just using your mouse. And we can have a look at one of the genes that is available. So if I would do a left click on this, then I would be able to find some metadata of this gene, where it is located, the size of this gene, a description, attributes, the name of the data set that it came from. And I'm also able to extract the sequence. So starting from ATG all the way to TAG. You can either copy paste it from here or you can download the FASTA directly. Bear in mind that this is the genomic area. And if you only want the coding region, then you have to scroll down a bit to the mRNA, which is a bit shorter than the sequence of the whole genomic region. You can also have a look at individual exons. You can download individual exons, etc., etc. So there's already a lot of information by just clicking on the feature. If you do a right click on the feature, then you have a lot more functionalities that you can use. So by view details, we will see the screen that I just showed now. But there's also the possibility to highlight, for example, this gene. So if you're working in a very large window and you want to focus on a specific area, you can also just highlight it. You can add it to the workspace. You can copy the sequence to your clipboard. You can download the sequence, send it to BLAST, use it for search, and also have a look at the gene passports. So in this case, gene passports have been made for, I think, both the genomes that are available for all of you. So there has been some extensive gene annotation going on. So if we look at this specific example, we can see that it is a nuclear transcription factor. It's located here. This is the taxonomy ID of this specific weed. We have the coding sequence here. We have the protein sequence here. The predicted localization. And if you're unfamiliar with what that means, there is a small description that you can hover over. So, well, it's pretty obvious in this case that the predicted localization is nuclear and not necessarily cytoplasmic. We have Interpro results, which is a public database, and it also contains links to Interpro. So you can easily go to the public databases if needed. We have BLAST annotations as well. So there's BLAST information in here. Go accessions from the Go database, and also here it describes that it is likely to be present in the nucleus, has something to do with transcription, etc., etc. So you have different types of data sources that will tell you what this specific gene/protein might do and where it operates. There's even some RNA-seq data available for this specific gene, but this is just test data to play around with the functionalities. And if we would have Variomics data available, for this specific genome, then we would also see a box with variant information here for just this gene. And that allows you to easily look whether you find interesting SNPs or variation for certain lines. So if we go back to the main window and have a look at the comparative map viewer, well, there is no genetic map information available for this specific genome. Doesn't necessarily mean that it's not there. So, I mean, if you find relevant information from the public domain, you can always ask the administrators to make that data available in KeyGene as well. So bear in mind that a regular user cannot add anything to the system. It can only visualize data and interrogate the data, but making data available can only be done by an administrator. We have the Variomics viewer here, and this looks different than what I just showed in the presentation, and that's because the WeedPedia already works with the new version of the Variomics viewer. However, there's no data yet available, so it's also not something that we can have a look at now, but it harbors the same functionalities, although the visualization has changed somewhat. If we go to BLAST, well, you can, every single user can do a BLAST in KeyGene, and it's only available for that specific user. So you can either type in a sequence or paste a sequence in here, define what type of BLAST you want to do. You can check some of the BLAST options, and you can also define to what database you want to do your BLAST. But also here, there's no publicly available BLAST information yet. The last part is the omics analysis, and I did not share that in my presentation, and that's because this is quite new in terms of functionalities. And recently there has been some test data put into the system. However, this test data is still fairly limited. So at the end of section 3, when there's still time left, I will show a video about all the RNA-seq features and functionalities, because it's a lot, and by going through the test data, you won't get all the essential information that you might want to know. Lastly, you can make a workspace in the WeedPedia. Again, it's just for you. However, later on, you can share the workspace with other people that have an account in the Pedia. So by just creating this workspace, I can store all the scientific research that I will be doing. So you can edit text, you can paste pictures in there, and all the data types or all the data in KeyGene that you might find interesting, you can store it to this workspace, and it will be visualized here. And later on with the RISE demonstration, we will have a closer look into the workspace. So there was one other genome available already, publicly available, horseweed, and also here there are only gene passports available. Well, not only, I mean, gene passports contain a lot of information, but there's no variomic data yet. There's no genetic map information yet. But I'm pretty positive that that will still be coming or will be generated later on. So this will hopefully provide you some insight of what's already available in the WeedPedia. If you have any questions on the general concept of Wikipedia, feel free to ask them. If you have any questions about the data of the WeedPedia, unfortunately, I'm not the person that can answer your questions, but you probably know who can. So I see that went by very quickly. So already, well, 20 minutes ahead of schedule. So that means that we have quite some time for questions. So yeah, go ahead.
All right, so if anyone in the room has a question, we'll come around with a microphone and you can ask your question as well. We're watching online for any questions to come through. And as you were seeing, we've been working on with KeyGene to upload these genomes that are publicly available so far to get them in. And Eric Patterson and Nathan Hall at Michigan State have been working really hard to develop these gene passports, what you're seeing. So there's an incredible amount of information that's already been searched and compiled from different databases that comes up at in one click when you're looking at any single gene. So that process, they've been spending several months to get that together, get it working, and with the KeyGene team to have that all integrated. But now that that's working, as you can see for kochia and for horseweed, then now there are a number of additional public genomes that we'll bring into that database, as well as the other genomes as they get finished. So that part is really exciting. And but yeah, on this first part, there's kind of the BLAST functionality, the gene passports, or if there's anything there, if you think there's some information you'd like to have when you look at a gene page that wasn't there, have suggestions for what to add. All right. Yeah, so sorry, we do need, I'm wondering, Carlos, could you help us out? If you could go to the back and grab a microphone, please. And then if you can run it around to the people asking questions. Thank you. Appreciate that. So Koen will just have a microphone here in the room, and then they'll go to the person asking the question so that you can hear it.
Sure.
Hey, does it work?
Yep.
Okay. So is it only going to be public genomes, or is there sort of like an incentive for people to send you genomes that they are working on?
Yeah, so I mean, Wikipedia is suitable for public genomes and proprietary genomes. So the administrator of the WeedPedia basically has the capabilities to assign data access. So whenever one of the partners has a specific genome that they want to add into the system, but should only be visible for them or maybe a certain group of people, then the administrator can make sure that that data only is visible for the persons that have the proper rights to see that data. So there is already a lot of proprietary data in there, but of course I can't show that to the general public.
Thank you.
I would add to that we do have a list of genomes that we're planning to put in the WeedPedia, and Sarah Morin's talk tomorrow will go through the list of there are a number of published genomes that are out there now that are good enough quality to put into the database. And then the consortium is working with a number of people who have projects in progress to maybe help out a little bit to get it finished to a certain level and then post it. There are various groups that you'll see who are working on genomes independently, and they've agreed once it's finished they'll make it available so we can put it in here. And then this the ability to log into WeedPedia will be available to anyone who wants to be part of the consortium. So just basically, do you have an interest in weed genomics? We'll set you up with an account. It's free to have that access. And then as Koen was saying, we are doing 10 genomes through the consortium. And again, in Sarah's talk tomorrow, you'll hear more about those. But the founding members, who are the sponsors of the consortium, have access to those for 1 year first. So the data administrator will designate that they can see those for that first year, and then once that year is up, then they'll be available for everyone to see. So, and we also have a question online from Tan with BSF. What other weed species will be in WeedPedia soon? So I'd say, Tan, yeah, we'll cover that in detail tomorrow, but we are anticipating that by about 18 months from now, we'll have 34 weed species in the WeedPedia, which is pretty exciting when you think that the Coniza canadensis genome was published in about March 2020. So we're going to have a nice inflection curve here in the number of genomes coming into the database. But another part of the consortium is to give the ability for people to just communicate, research networks, update on what's going on, because there are certainly people out there who are working on a weed genome that maybe the consortium maybe doesn't know about, and we want to work together, or you want to host it in here. That's definitely an option. All right, any other questions in the room on this first part? Koen, I have a question on the data management. So if we go to the hamburger menu, that's where for the genomes to which a user has access, they can actually download the FASTA file, right, if they want to download that genome themselves?
Yep. So yeah, that was a sort of spoiler, because that was intended at the end of the last section. But it doesn't matter. I mean, we can also do it now. So yeah, all the information that a user has access to can also be downloaded. So I can imagine that when you get to work with the WeedPedia, that certain data sets that were already in there, that you might not have these available locally. So in the hamburger menu, there's indeed a button, data management. You will see again these 2 genomes that will be later on a very extensive list. And you can have a detailed look to what types of data is present for downloading. So here, for example, it says that there are over 612,000 mapped features that you can download. So that's the genome annotation that's present there. There are BLAST databases that you can download. So, I mean, you don't even have to make your own BLAST database if you want to do stuff locally. There's RNA-seq information in here, and also for the other genome, again, genome annotation. So if you would enable one of these features, you will find some metadata about this data set. So in this case, I selected the mapped features, and it says horse wheat, number of files, taxonomy ID, what type of file it is, so it's a GFF, and the source where it came from. So if I would now click on this GFF, you can already see at the bottom of my screen that you will be able to download the file. I'm not going to do that as an example, but believe me that it works. So that's the way of downloading information from Koepedia and use it locally if necessary. Something else that we that you can also do. There is an extensive API present in Koepedia, and not everyone will be familiar with APIs or how to use them. But basically, by making use of URLs, you can download files or retrieve data from the system without you having to browse to it. So if you are interested in using the API, and there is a very extensive manual available that basically shows you how to access data via URLs and what the output of that is. And you need access tokens for that, but by then the Koepedia administrator can of course provide that for you. So there's really a lot of functionality available to basically do everything in Koepedia, but then by navigating through URLs. Does that answer your question?
Yes, thank you. Do we have any other questions here in the room or online? Again, you can use the Q&A link that's in the lower left-hand corner of Chime, and we can read those questions out. All right, well, I'm not seeing any more questions here in the room or online. So do we want to maybe take just a little bit longer break then?
Yeah, that sounds good. I mean, okay. There's a possibility to start the RISE demonstration earlier, but yeah, if people were waiting for that and they tune in at 9:45 or at 10, then obviously they miss a bit.
Yeah, how about let's do that. Let's plan to we'll come back here as scheduled at 9:45, right? Mm-hmm. I think that's when session 2 starts.
No, it starts at 10.
Oh, it starts at 10.
Yeah.
Okay. Yeah, but we probably don't want to. If people are following the schedule online, they'll be that. Well, that'll give us a little time here to have the coffee break. And is it possible? Oh, yeah, go ahead. Okay. Well, I think that's probably what we want to do. So the next session is scheduled to start at 10. Mm-hmm. And then and that's going to be covering you go into more detail on I forget which sections are we covering for that part?
Yeah, so the next session will be a demonstration of Koepedia with a use case of RISE. So there we have a very precise scientific question that we want to address, and then we will see how Koepedia can answer that question.
Okay, excellent. Well, that does make me think of maybe one question, and just to show everyone some of the custom search features that we've put into our gene passports. Mm-hmm. Could you show them, if you could share your screen in WeedPedia with the Bastius-Goparya genome and show if you type, for example, EPSPS into the search term. Mm-hmm. To look specifically.
I will do it for me.
This is one of our genes that people tend to look at quite a bit.
Let me see if we have. Yeah, we have it enabled. Okay. So here's the search result for EPSPS.
Yeah, and then maybe if you click on the gene passport and you can see.
Yeah, so then we end up indeed at the window that we already had a look at before.
Yeah.
And indeed, from there you can again go to the Genome Browser or add to the workspace, etc., and have a look at what this apparently very important gene does.
And if you could go back up to the top, Nathan and Eric put in some custom terms just so everyone knows. So you can see HRAC:9. So that's of course the herbicide group for glyphosate. So if we have a known target site gene of a given mode of action, you can type in HRAC: and then that mode of action number. And if we've been able to annotate that target site gene in there, then you'll be able to find it. So that will work for ones where we know, if we have one or a few target site genes. You can see also glyphosate, EPSPS. So some of the common kind of abbreviations, terms that people in our field would use. So this would work for ALS, HRAC2, for example. So we certainly don't have every single herbicide, but I think at least as far as mode of action group, that can help you navigate to find, and that's in the pipeline to annotate that for every genome that's coming through. So that's pretty exciting. And they're also working on a pipeline to annotate anything that would be, for example, a cytochrome P450. So you can just type in P450 and you should get a list of everything that is predicted to have that function. Of course, with those, you have to do a lot more manual curation. The same for GSTs. So really, we're trying to build into the annotation some of the typical things that people would be looking about when thinking about herbicide sites of action. Yeah, I've got another question here.
Sorry, I was going to wait until after the next section, so I may be jumping the gun a little bit here, but the herbicide target gene thing made me think. So if you upload a genome that's not annotated, can you compare across genomes and try to align and find these genes that are annotated in another genome?
Mm-hmm.
Yeah, it's an excellent question. The answer is yes. However, you need a bit more than just KeyPedia. So to recap first, KeyPedia is not a data generation tool. It's a data visualization / interrogation tool. So if you would have a blank genome in there, then you will need bioinformatics capabilities to make, for example, a synteny analysis between these genomes, or by mapping features of a particular genome to another genome. And then these data sets that basically contain the links between two genomes or multiple genomes, those can be uploaded into KeyPedia, and those can then be visualized, and then you can exactly see that if you have genomic annotation from genome A and you then add a track with mapped data from genome B, then you can see exactly whether genes are present there, yes or no, or whether there are base differences or structural variants. So yes, it's possible in KeyPedia, but you need to do the analysis up front.
Okay, thanks.
And later on in the third section, we will go into extensive detail on what types of data can be put into the system, so that you know how to generate the proper data files for KeyPedia. Basically, KeyPedia can handle every common format. However, as soon as you deviate from that format, then KeyPedia will not accept your input data.
All right. Any other questions? I'm not seeing any more questions online still. Well, feel free to take a minute to, during the break, think about the KeyPedia, and in the next session, starting at 10 a.m. here Central Time, we will get more into some of the basic and intermediate functionalities of WeedPedia. And as Koen said, we'll look at the rice genomics data. So then you can really see, this kind of helps everyone think ahead. What can you do? What kind of data can you generate that can then go into the WeedPedia, and what can you do with it? So thank you very much, Koen. So yeah, we'll just have about a 30-minute break here then, and come back at 10 a.m. local time. All right, thanks everyone.